terrific. Good morning, everyone, and thank you for being here. Thank you, Dave, for your presentation. It's a wonderful backdrop, I think, to this whole event, and you'll see a number of echoes of what Dave has said in my presentation today. But what I want to focus on really are the soil biota. Who are they? What are they doing there? What might we be able to do to encourage their beneficial activities and discourage ones that we are not so happy with? So this is the outline of my talk today. I'll emphasize the importance of the soil biota, talk about energy and organic matter formation and transformation. I'll introduce you to the main groups of the soil biota, and then finish up by talking about a few hotspots of biological activity in the soil, and then sum up from there. So this slide should be a bit reminiscent of what Dave Wolf has just said to you. It's organized in a slightly different way, but I think that it provides for us the same sort of information about why the soil biota are important for soil health. So first off, we understand that organic matter going into soil is going to form the foundation of the soil food web. And we know that the soil biota are very important in transforming these residues through their decomposition processes and through their nutrient cycling activities. We also know that as residues decompose, we start to form aggregates, which are a very important portion of soil health, as it allows both air and water flow through the soil. And we can see here that water infiltration, water flow, water storage are all affected by these activities. The soil biota also can produce things like phytohormones and other plant stimulatory types of compounds, and thus lead to plant growth enhancement. Other things, I think Dave mentioned nitrogen fixation in the rhizobia, and I will come back to these a little bit later. But other things we have are things like pathogen suppression and other modes of plant protection. And then lastly, things like detoxifying pollutants here. So we know that the soil biota influence all sorts of facets of soil biology, of soil health, excuse me. All right, so I wanted to start with this concept here, and this is one of my favorite cartoons from Gary Larson. And I think that it helps us understand really what drives the below ground ecological processes. And we can see here early chemists describing the first dirt molecule. We can see here DE, and here's the molecule itself. But what I want you to focus on are these two little lines right here. And some of you may recall back in your chemistry days what these two little lines represent. And unfortunately, I would ask you at this point, and if anyone wants to unmute and answer this question, I'd be delighted for you to do so. But we understand these two lines here to be bonds. And in these bonds is represented chemical energy. And it is this bond energy that most of the soil biota in a group that we call chemoheterotrophs rely on for their energy, being able to garner energy in the soil. So this is very, very critical for survival and function in the soil environment. Okay, so when it comes to biological activity, I wanted to discuss these two primary limiting factors, because this is how I kind of organize my thoughts about biological activity. I asked two questions: where is the energy coming from, and where is the carbon coming from? Because both of these things are absolutely essential for life. So we know that energy supply for things like plants is going to come from light. And so light penetration is going to affect the ability of plants to be able to garner energy and photosynthesize. And from that point, plant litter coming onto the soil surface, rhizosphere deposits going below ground become the substrates that are available for soil organisms. And so that energy that's captured in light goes into plant biomass, and then the products of plant production become the substrates for soil organisms, so that we have an energy piece to this. The other part is, where is the carbon coming from? And we know that for plants, this is through the process of photosynthesis, and that plants are using carbon dioxide and fixing this into sugars, again, becoming the organic carbon that then the rest of the soil food web relies on. So much of the biota in the soil are going to rely on these photosynthetic products. We have a few key organisms in soil that are also able to fix carbon dioxide, but these are much fewer in number, and only a few are highly critical. So if we start here then, looking at that energy capture and carbon fixation piece, which is where all of the carbon in soil comes from, which is what drives the whole biological process. So here we have the sunlight, CO2 and water. Through photosynthesis, we have the production of oxygen by plants, and then sugars being produced. And so from here then, the capture of that sunlight into photosynthetic products down into the rhizosphere then becomes that energy and carbon piece that is so critical to the function of the soil community. So from here then, organisms start to participate in nutrient cycling processes, soil aggregation processes, some plant protection activity, detoxification, and more long-term stabilized organic matter formation. So if we pull back out from this and we look, here's our sunlight, here's our photosynthetic activity, there's our sugars going into the rhizosphere. This then supports a whole community of organisms below ground in this region that we call the rhizosphere, which is one of several biological hotspots in soil. And so this wonderful slide from Jennifer Moore-Kucera at the NRCS gives us this flow of energy into this soil food web that has a variety of different organisms, as we can see here. So if we focus in on this soil food web piece, here's a much simpler diagram of what we mean by the soil food web. And many of you would be thinking about things like food chains. You have your autotrophs producing, your plant producing communities, and then you have your consumers and your predators and all of this. And we have all of these types of functions represented in the soil community as well. So again, sunlight coming in through photosynthesis, the production of plants and the rhizosphere community, organic matter coming into soil. And now this is what is going to support the rest of this soil food web. So in this first trophic level here, these are our primary consumers. And so who we have in here are a variety of soil bacteria, a variety of fungi, different types of nematodes. Now in this primary consumer category here, these nematodes are going to be root feeders, feeding directly on plants. So these guys are not what we really want to see within the soil system. And so it is incumbent upon us to figure out ways to really bolster these other nematode communities in the soil and to suppress root feeding nematodes overall. Also at this first trophic level, we have earthworms. And I'm going to describe each of these different organisms and talk about their importance in terms of processes below ground. So these are our primary consumers. Now in our secondary consumers, we start to see things like protozoa coming in, other types of nematodes that are not plant parasitic, and a variety of different types of arthropods. And we can move up from there into higher trophic levels with predatory nematodes and predatory arthropods, and then up to birds and other creatures. So we know we have an organization here, but it isn't so straightforward as it's not really linear. It's very, very complex. And so this word web is very important in the discussion here because these feeding relationships can become quite complex. Okay, so I wanted to start with the rhizosphere and talk about this as a carbon pump to putting organic matter below ground in the first instance. And so plants, once they photosynthesize, then translocate those fixed sugars around the plant down into the rhizosphere. And then we have a variety of different compounds that come out, things like exudates that are leaking more passively from the roots, secretions that are more actively produced compounds that are transported across the cell membrane, and then things like lysates as cells are ruptured and die and release their contents into soil. Now, all of these are very energy-rich compounds. And so what we find in the rhizosphere is a dense population of organisms right on the root surface, which we call the rhizoplane, and in the soil surrounding the root that is influenced by these carbon compounds coming out of the root. And so we see that we have not only things like sugars that provide energy, but we may also have other things like chemical signals that then are received by particular organisms in soil and then begin a chemical dialogue crosstalk between them. And a good example of this would be with rhizobia, where the root is producing signal molecules that are received by the rhizobia. The rhizobia themselves produce signals that tell the plant, hey, I'm here. And then we have an orchestrated set of things that happen after that that ultimately culminate in the formation of a legume root nodule and the fixation of nitrogen from the atmosphere. So these chemical signals are also extremely important component of what goes on in the rhizosphere. So we see that we can have a variety of organisms, and we've talked about it in terms of the food web, but we also have got the activities that they engage in. So things like the production of various enzymes, growth regulators, hormones, antibiotics, et cetera, that can influence plant growth and production. So we have energy being garnered from the plant, and then the organisms in the rhizosphere then communicating with the plant in different ways and helping with plant growth and development overall. All right, so I want to switch to talking a little bit more in detail about who is actually there, and I'm going to start with the bacteria. Here you can see bacteria colonizing the head of a pin. So we can see that they're extremely small and not very self-descriptive. So we have rods and we have cocci and we have spirals and these sorts of things. But it's very difficult from just looking at an organism to understand exactly what it is doing there. And I think Dave referred to a lot of the molecular biology that is going on now to try to identify organisms in the soil and rhizosphere and understand what their roles are. But bacteria overall are responsible for organic matter decomposition, and through that process leading to the release of important plant macronutrients. The thing to remember is that through the process of decomposition, what are these organisms trying to do? They're trying to feed themselves. They're trying to grow. They're trying to reproduce. They're interacting all the time with other elements in the soil. So they're not actually decomposing for the express purpose of feeding plants. They're basically doing this to help themselves. And in the process, what is left over then becomes available in the soil solution. So if nutrients are limiting, often we can see that bacteria and fungi can actually retain these nutrients and immobilize them in their biomass for a short period of time. This can be beneficial in some ways, keeping those nutrients in the soil and keeping them from leaching. But under conditions of low nutrient availability, it leads to immobilization and then to reduced nutrient availability for plants. So we need to balance these various things. Other things bacteria are able to do are to control pathogens. Many of them are pathogens. And so looking at the balance between these two activities is also part of the study of soil health. Other things bacteria can do that we are very interested in are things like solubilizing phosphorus. This can be quite important in soils where phosphorus fixation is a problem. They are also responsible for primary aggregation of soil through the production of bacterial metabolites, which tend to be sticky kinds of substances, which help with that primary aggregation at the clay platelet, above the clay platelet level, and lead to the formation then of microaggregates. So they're very, very important in this formation of the organo-mineral complex, as we call it. Okay, so bacteria are extremely important in nutrient cycling activities. And here I give you the nitrogen cycle. It is a fairly complicated cycle, but one of the more intriguing things about it, is that it is dominated by microbial activity. At every single step, we rely on bacteria in some way, shape, or form to transform nitrogen into another form. So if we look at atmospheric nitrogen here, the biological nitrogen fixation is the exclusive domain of rhizobia type bacteria. Once we have biologically fixed nitrogen in this organic nitrogen pool, this needs to be decomposed. So once this is decomposed, that mineralization process is very much dominated by both bacteria and fungi, but largely by bacteria, into this ammonium pool. Now the ammonium pool is available for plant uptake, but we also see that bacteria are critical for the transformation of ammonium into nitrate, which is also one of the pools that plants take up. From nitrate, we can have denitrification, again, another bacterial process, reconverting that nitrogen back into atmospheric nitrogen. So bacteria dominate this cycle. So we need to be very aware of what's going on with them if we want to be able to manage nitrogen efficiently within the soil system. Okay, so in terms of nitrogen fixation, this is one of the very successful interventions that we have had in agricultural production over the last century. So in the early 1900s was the first distribution of legume inoculants. These organisms weren't really even discovered until the late 1800s, but we recognized their importance fairly early on and started developing inoculants in the 1920s and 1930s. And we still do use these today. So you can see here, this is an example of a bean that is not inoculated. This one here is inoculated with a peat-based inoculum, which is why it is black. The bacteria are living within that peat that is put onto the seed. And then we can see the response here to inoculation in this. This is a Hawaiian oxazole, very nitrogen deficient. So we can see without the rhizobia, the plants are really struggling. With rhizobia, we get a lot of nitrogen fixation in here. The rhizobia form these nodules on the roots of plants, and that is where nitrogen fixation takes place. So we are very beholden to this relationship to garner nitrogen from the atmosphere, recombine it, and make it available to plants, and then available again within the nitrogen cycling system. Sometimes we get a response to inoculation, sometimes we don't. We can see in the foreground, these are cowpeas right here. And I have 3 different treatments in this field trial, which are more easily seen with the bean plants that are right here. There's an uninoculated, inoculated, and a nitrogen fertilizer control. And we can see very clearly for beans, we have a nitrogen deficiency. We get a good response to inoculation, and we get a good response to nitrogen. We can see in this back row with soybean, again, nitrogen deficiency, good response to inoculation, good response to nitrogen. But we're seeing nothing here with cowpeas. So what the heck is going on with cowpeas here? Why are they not nitrogen deficient? And it turns out that here at this field site, there are plenty of rhizobia in the soil that are capable of nodulating this plant. And so this is something that we need to think about. We develop complex inoculation programs, but I think we need to often stop and ask the question, do we already have a sufficient population of these bacteria in the soil in order to satisfy the demands of the plant? So understanding this about rhizobia can help you decide when and where to use inoculation to the best benefit. Okay, other things that we can do towards managing organisms in soil are other types of inoculants. So we have nitrogen fixing bacteria. We can have phosphate solubilizing bacteria or others that produce phytohormone analogs. We often see out there on the shelf these microbial soups, and they're not something that I'm willing to recommend at this time, but they are out there. They contain plant growth promoting rhizobacteria. That's what this means right here. But often it's just a mixture of different bacteria that have been shown to do various things where the product itself doesn't actually have a track record. So I'm going to warn you a bit about just jumping right in and buying microbial soups. Other things that can be beneficial are things like compost teas or the use of signal molecules alone. Other things that are extremely helpful for being able to encourage your bacterial community fall under the rubric of cultural control. Things like adding organic matter. Why would organic matter be important in terms of trying to bolster your bacterial community? Well, it's food, right? It's food, it's carbon, it's energy, and this is what's going to drive that below ground community. So yeah, feed me, give me roots, give me organic matter. Reducing the use of toxins within the system, keeping plant roots in the system. And I think that Dave made a very nice plea for cover crops, and I know that Matt is going to speak about those tomorrow. But keeping roots in the system, you're pumping carbon below ground all the time. So I think that this is a very important concept within the whole framework of soil health as well. Other things, oxygenating the soil, improving drainage, lowering bulk density, all of these things are going to be beneficial for your bacterial community. Okay, so soil fungi now are quite different organisms from bacteria. Bacteria are basically single-celled, fungi are multicellular. We're moving from a prokaryotic to a eukaryotic organism. And fungi grow in these long tube-like structures that you can see here in this agar culture. These are called hyphae, and they continue to grow just by the extension of the tips of these hyphae in soil. And so you can see that they can grow into all kinds of little crevices and things, and they can expand and cover great areas. I think in England, they've mapped one fungus, one genetically identical fungus covering more than 4-kilometer area. So these things can really make tracks. So that's what it looks like in agriculture. When you see them in the soil in large amounts, you'll see this fluffy, cottony material. Those are just the hyphae in large quantities. And when we see them this way, we call them the mycelium. So fungi are also critically important in the decomposition of organic matter. They're important in mobilizing phosphorus, particularly with a group of fungi called the mycorrhizae, which I'll speak about more in a few minutes. Fungi can also help to control pathogens, promote plant growth, again, by the various compounds that they can produce, including phytohormones. We have some that are able to infect and control insects like Metarhizium and Bovaria. And then we find that fungi are also really essential for macro aggregation and soil stabilization. So just looking at a few more pictures of them here, we see a mycelium decomposing this organic matter on the soil surface here. We can see very clearly the hyphal strands holding this bit of organic matter together. And this is actually a fungal hyphae ramifying through a leaf as it begins to excrete exoenzymes and decompose this organic matter. And then lastly, we see them starting to reproduce here in the soil. So one particular group of fungi that are critically important in agricultural systems are these what used to be called vesicular arbuscular mycorrhizae. It turns out not all of these, the fungi in this group can produce vesicles. So this older name has been supplanted by AMF, arbuscular mycorrhizal fungi, AMF. Excuse me. And here we can see a plant root. We can see the fungal hyphae on the exterior of the plant. But what is unique here is that these fungi actually infect the cortical tissue of this root. And in their interaction, this is actually an infection inside a cell. And this is what is called the arbuscule part of this fungus. You can see arbol, tree, they're little tree-like formations within the cortical root cells. And it is here that is the site of transfer of carbon from the plant to the fungus and of phosphorus and other poorly mobile nutrients to the plant. So that is the importance of the mycorrhizal fungi, is this transfer of poorly mobile nutrients from various places in the soil to the plant root, and the exchange of those nutrients for carbon to support the growth and activity of the fungus. So these guys can be seriously important in a variety of different contexts. The arbuscular mycorrhizae, there are 5 different groups. The 2 that are most common in agriculture are the endomycorrhizae and the ectomycorrhizae. The ectomycorrhizae now are in a slightly different group than the arbuscular mycorrhizal fungi. They tend to infect conifers, and we can actually see here a small pine, and we can see its roots coming out down through here, but all of this material here, this haziness that we see in the soil profile, those are all hyphae extending into the soil. And so I think this is a beautiful photograph that really shows us how that fungal hyphae, how those fungal hyphae can extend into the soil. They can take up nutrients, mobilize them along their hyphae, move them to the plant root, and then exchange them for carbon. Thus, the fungus benefits by its relationship with the plant, and the plant benefits by obtaining poorly mobile nutrients. Okay, so some things we can do here to help them along is to use various inoculants. For the ectomycorrhizal fungi, these are fairly easy to make and use. For the arbuscular mycorrhizal fungi, one trick there with them is that they are obligate plant symbionts. So consequently, to make an inoculum for them, you have to grow the organism with a plant and then use the plant roots chopped up or the spores that are formed as your inoculum. It's a much more difficult process. But other things that we have, trichoderma that Dave mentioned before, and then the insect infecting types that I mentioned before. Other things we can do to help fungi are in the means of cultural control by reducing tillage, adding organic matter, reducing toxins, and keeping plants in the system. That is particularly important for the mycorrhizal fungi. Okay, other things we have in there are earthworms. We can see here that they are processing a large amount of organic matter here. They're also drilling into the soil and forming these channels and producing casts at the surface that are high in processed organic matter and in microbes and are very helpful for soil fertility. So they reproduce by means of cocoons that we can see here, and we have some potworms also illustrated on here. We do have vermiculture that goes on. There is at least one company operating in New York State that produces vermicompost for use in largely in horticultural production. But earthworms overall, we can say they help to stimulate microbial activity. Through their processes of burrowing and casting, they're mixing and aggregating soil. As a consequence, we can see increased infiltration, channels for roots, and the burying and shredding of plant tissue, which then helps to increase the decomposition kinetics of this material. So how can we help them out? Some cultural control, reducing tillage, reducing chemicals, and increasing organic matter inputs. Okay, so overall, if we think about this role of organisms in the soil, I wanted to make this point that as the complexity of the food web increases, we have a greater release of mineral nutrients. So here we see a sterile soil, and what shoot biomass is at 77 days, it's quite low. If we add bacteria that can then process that organic matter and lead to nutrient release, we get a big boost in plant production. But if we also add in higher order feeders that are now feeding on bacteria or feeding on fungi, then we get a greater release of plant nutrient elements and a greater plant growth. So this comes about through the activity of higher order feeders, largely the protozoa and nematode communities. So what I've shown here are a few different types of protozoa. We have the ciliates. Here's a ciliate feeding. We can actually see the cilia here beating. There are bacteria out here that it is moving towards its mouth parts right here, and then into a gullet and into a digestive vacuole. We have other types of flagellates. We have amoeboid types. Hopefully you guys are familiar with this. And then we also have testate amoebae. This is a test right here. The amoeba lives inside this test, which helps to protect it from environmental harm. And we often see tests formed in more in acidic soils, in acidic forest soils in particular. We see a lot of testate amoebae. So we have a diversity of different protozoa. Some feed on bacteria, some feed on fungi, and some feed on organic matter. So as far as their functional roles are concerned, we see that they consume a lot of bacteria in soil. A protozoan can consume up to 30,000 bacteria in a day. In so doing, they regulate the population size, they accelerate the turnover of organic matter, and they help to maintain plant available N. Lastly, they can help with pathogen establishment. Some things we've used protozoa for. Now, protozoa are single-celled. They do not have a cell wall, so they tend to be quite sensitive to the environment, and consequently, they've been used as an indicator for hazardous waste. Other things, they tend to be a food source for some fungi, nematodes, and a variety of other organisms. And lastly, we do need to recognize that some protozoa can cause disease, so it's not all of a benefit. So one thing with the protozoa and with the nematodes and this higher order feeding is an increase in the amount of nitrogen that can be released into the soil. And this comes as a consequence of the carbon to nitrogen ratio of the material they're feeding on, but also of their own bodies. What is it they're trying to maintain in their own bodies? We use this to estimate the likelihood of mineralization. And we start with a few basic concepts, and I'm not going to go through all the details of this, except to say that bacteria tend to be very nitrogen rich, fungi less so, and protozoa somewhere in between. So I want you to think about a protozoan that only needs 1 nitrogen for every 10 carbons, who is eating 30,000 bacteria per day that have 1 nitrogen for every 3 carbons. There's going to be a lot of excess nitrogen that will be released by protozoa and nematodes that are feeding on these nitrogen-rich bacteria. And that, in a way, is how we get inorganic nitrogen re-released into the soil. Okay, other things we have out there are nematodes. These are vermiform or worm-like looking, but quite a bit smaller, up to 0.5 micron. We find they're ubiquitous in soil. They depend on water films, and they feed on a wide variety of different organisms. So when they are plant parasites, they're very clearly a problem for us, but there are so many other nematodes. And this whole process of feeding and turning over the microbial biomass and re-releasing nitrogen is an important function of these soil animals. We have a variety of different types, as I said, feeding on a variety of different things. Dave showed us a great picture of one bacterial feeding nematode, but we can see that nematodes have all kinds of different mouth parts, and based on what their mouth part is, we get some clue as to what they may be feeding on. The other thing that we look for within nematodes is whether they possess a stylet or not. And you can see a stylet here in this picture. This is a piercing organ that is then shot out through the mouth parts and used to pierce either fungi for fungal feeding nematodes or plants for plant feeding nematodes. So for the functional roles of nematodes, then again, we have them feeding on bacteria, fungi, and protozoa. Also plants, which is a problem for us, but the bulk of them are actually beneficial for us, help to control bacterial numbers, release a lot of nitrogen through the feeding process, as I described before, and help to maintain plant available N. So some of their selective feeding can influence soil structure, carbon utilization rates, and the substrates that we find in soil. So they can be quite influential. And again, I need to mention that some of these can be important parasites, not only of plants, but also of animals. So some things that we can do to control things like plant parasitic nematodes are to use different types of entomopathogens, and there are inoculants that are available for this. Also biocontrol types of processes. But in fact, our best bet for controlling plant parasitic nematodes is to encourage a diverse soil food web. Okay, so in a gram of soil, there are billions of bacteria, millions of fungi, and thousands of nematodes and protozoa. So we can understand the soil system as really teeming with life. When it comes to how many different kinds, that can also be a staggering number. We can have as many as 10,000 different types of bacteria, 5,000 different types of fungi, and hundreds of different types of nematodes and protozoa. So it's a very complex system. And I think, as Dave mentioned, as we move into the molecular era, we're starting to understand more and more about this system. That is leading us to be able to manage it better. Okay, I wanted to move on to talk a little bit about the soil fauna. So moving now up to small soil animals. Here we have Collembola as part of the soil fauna. And we also have here a pseudoscorpion that is actually eating one of our Collembola here. So the soil fauna are very important for us because they perform these various functions. Shredding of organic material is very important. As it comes in and it starts to be shredded apart, the surface area of that material increases. As the surface area increases, we can get more colonization by bacteria and fungi, and then speed up decomposition and organic matter turnover. So this then stimulates microbial activity. Some of the things that they do can help to mix microbes with their food. They're an important part of this mineralization of plant nutrients through their shredding activity and through their waste products, also known as frass. They also help to enhance soil aggregation. Some of them may burrow, some may control pests, and some may also be pests. So again, a very important component. Here's a few examples of shredders. We've got the dung beetle here, sow bugs, and a mite. And I think this photo of the mite makes the shredding point very nicely, because we can see that it's sitting on a leaf, and we can see that all of the material in between has been shredded away. We also have got fungal and litter grazers, again, turning over this microbial biomass, re-releasing nutrient elements into the soil. And most of these are pictures of Collembola, except one right here, we have a sow bug sneaking into this grazer classification. Others within the soil fauna, we have herbivores, which of course are the ones that we want to control, things like simplins, and mole crickets, and other grazers. This mole cricket is quite an amazing thing. I saw one for the first time when I was in Kenya, and they're several inches long, and they can decimate a plant in a day. So finding ways to control this component of the soil fauna is very important. Others that we want to encourage are various soil predators that can help to control crop pests, excuse me, things like pseudoscorpions, wolf spiders, and harvester ants. Okay, so that's sort of an overview of who's there in the soil. I wanted to briefly touch on a few more hot spots of soil and then begin to sum up. Okay, we talked about the rhizosphere as being a hotspot of microbial activity in the soil, and the reason for that is that we've got movement of fixed carbon going below ground, creating that hotspot. But another hotspot where we have a lot of carbon is in the litter layer. And this is what we call the detritosphere. Here we have a lot of our fauna are going to be living in this litter layer. So it helps to protect the soil, conserves moisture, lowers temperature, and we have this great carbon source for organisms. And so who we have there are a lot of the mesofauna, things like springtails and mites we just had a look at, and others in the macrofauna, beetles, centipedes, ants, and isopods. So the detritosphere is another hotspot. We also have to think about the pore space in soil and to think about aggregation more largely. So an aggregate or soil, generally speaking, is going to be have a mineral component to it, and it will have a pore space. So we have minerals and we have pores, and those pores are variously filled with water and various gases. Now, when those gases are only carbon dioxide, we've got an anaerobic situation going on. We want to have oxygen in those pore spaces. And so they really are this lung and circulatory system of the soil are these pore spaces. We need to have good air infiltration. We need to have good water flow. We do not want water saturation conditions because again, leads to anaerobic conditions, inefficient turnover of organic matter, and basically slows down the whole system. So the pore space is another important area within the soil. And lastly, we have the aggregate space or aggregatosphere as another important place where organisms are contributing to overall soil health. Aggregates create stability and resist erosion, protect organic matter, but are a source of energy and carbon for organisms overall. So they help to create the pore space, and they are created by the microbes that contribute to their formation. So organisms are very important in the chemical stabilization of soil aggregates through their metabolic byproducts, as I was talking about before, many of which are polysaccharides that help to bind soil particles together. We have other compounds that are produced by organisms in soil, such as glycoproteins, which also help to bind soil particles together. So we have a huge contribution of organisms to the stabilization of soil aggregates. Okay, so I'm just going to sum up here in a couple of more slides. I recognize I only have a few more minutes left with you. So I wanted to re-emphasize a few things. So microbial activity rates and what affects microbial behavior in soil come down to chemical, physical, and biological factors. We need to be cognizant that all of these things are going to affect microbial activity rates. Things like pH, the distribution of oxygen relative to CO2 and how that may lead to anaerobic or aerobic conditions. Texture, porosity, compaction. Again, how well is air and water flowing through the soil? And then on the biological factors, things like competition, which will limit the activity of various organisms. Predation and allelopathy are other things to consider. Okay, so key processes in a healthy soil. We want organic matter decomposition, turnover, and accumulation. We want to have good nutrient transformation and availability. We'd like to suppress disease. And we want to be able to support this microbial community overall, because it in turn is going to support plant production. Okay, one thing to leave you with is to have a look at what's going on with your plants. Oftentimes people are looking out there, they're scratching their head, what the heck's going on? Dig it up, have a look. Roots are really, really excellent indicators of soil health. They can tell you if you've got poor drainage, poor nutrient availability, compaction, whether you have various and sundry pathogens. So by all means, have a look. They will tell you what their life is like if you give them a chance. Okay, so in summary then, soil is a very highly complex and dynamic living system. We need organic inputs for the energy and carbon to be able to drive that system. These organisms are sensitive to the inputs that we put in there. They're sensitive to tillage and pesticides and other things that we use. So we need to be cognizant of those things, limit their use wherever we can. And then to increase nutrient availability and plant productivity, we need to maintain this healthy soil food web. Okay, so that's what I have for you. I'm bang on time at noon. I'll leave it open for any questions. Joseph?
Yeah, any questions? We got a couple of minutes and Janice can be here. There's a lot of information. So if you're a soil biology enthusiast, this is a good opportunity to ask good questions.
Hi, this is Patricia speaking. I am curious. This is a pretty general question, so I hope that you get some kind of guesstimation. But is there an ideal ratio of fungi, bacteria, and protozoa that seems like, I know that according to the soil type and whatever ideal crop is being grown, but is there a general answer to that?
Yeah, so this is an interesting discussion within the soil biology community, is this fungal to bacterial ratio. There are some folks who really put a lot of stock in it and think that you should be driving your system toward more stabilized, what they consider more stabilized, which would be more dominated by fungi rather than bacteria. So often they'll talk about bacteria as being indicative of a rich, a lot of organic matter availability, so copiotrophic, well-fed kind of circumstance. And the thinking is you really don't want to be there. You want to be more towards stabilized organic matter, turning over more slowly and more dominated by fungi. That is the thinking. Now, there is some evidence for that, but then there are also folks who don't subscribe to that. As to an ideal number, I don't know that anybody has struck that, but the thinking is about the stability of the system, and that is pushing more toward more complex organic matter going in. So you're driving it more toward fungi than bacteria, but I'm not 100% convinced personally, but go ahead.
Thank you. Thank you very much.
Sure.
I actually wanted to have you clarify a little bit more about the carbon nitrogen rate. Hi, good to see you, Janice.
How are you?
I'm wonderful. But I'm in a space, I just came into the office, I'm in a space right now, so I have to keep my mask on. So I wanted to ask you a little bit more about carbon nitrogen ratios in
Yes.
terms of, so you had bacteria need a 3-to-1, fungi need 15-to-1, and protozoa need 10-to-1. So those are all like competing numbers. And my understanding is that, healthy soils have all of these things. So
Yeah.
if we're manipulating our soils to have more of one than the other, we're clearly influencing the populations. So I guess how do we work with amending soil or improving soil so that there's a balance here, so that we do have a good habitat for all of these decomposing organisms?
Okay, so I want to, this can be very confusing, and I took a slide out because it takes too long to explain what's going on, but there are two ways to look at this carbon to nitrogen ratio. One is the carbon to nitrogen ratio of the material that is going into the soil. And because you spent such a long time teaching composting, that somewhere between 20 to 35 to 1 is your break-even point for mineralization versus immobilization, okay, of being able to turn over that material or not. So you need to have enough nitrogen to turn over that carbon. So that's one discussion that we have. How many browns, how many greens go in, okay? That is a little bit different from what I was trying to explain here with the cellular ratios that organisms try to maintain. So bacteria are anywhere from 3 to 8 to 1. There's wide range, and often I'll hit on 5 to 1 because it's easy to talk about. Fungi need less nitrogen relative to carbon. And protozoa needs somewhere in between. They're similar to us, about 10 to 1, 1 nitrogen for every 10 carbons. So the point here is if you need 10 to 1 and you're eating 5 to 1, you're eating twice as much nitrogen as you need. So you're going to excrete that. So that's the discussion on C to N ratio on inside organisms, which is a different discussion than what we want for putting materials into soil. Does that help? So
Yeah, no, that makes that distinction is really clear,
yeah.
and really important to me. So thank you.
Okay. So everybody's eating all the time, and if we have a lot of nitrogen available, you're probably going to bolster your bacterial community relative to your fungal community because they can get in there, they can get it fast, they can turn it over quickly,
and they'll heat it up for you while they're doing it.
Absolutely,
Yeah.
Absolutely.
Just want to break the questions really quick to say, if you want to go, pretend like this Zoom meeting's over and then rejoin us at 1, get lunch, go outside. So yeah, if you want to split now, you can just rejoin us at 1, but also we can answer a couple more questions here if you have them.
Hi, this is Sam Joseph. You mentioned a bunch of potential inoculants that are available. Are there any that you highly recommend? Like, I've never heard of can't remember how you phrased it, but the phosphorus, like making more available to plants inoculants. Do you have any brands that you recommend, or actual like sourcing of this inoculant?
No, I don't have any brands to recommend. Right now, I have a student in my lab from Pakistan who's working with some of these bacteria, isolating them. So very soon we're going to have our own strains. But I have been down to the local Green Tree, has got a few formulations down there that will include some of these phosphate solubilizing bacteria. Basically, what they do is they produce phosphatases that can release phosphorus from the soil complex. That's kind of what's going on there. So the inoculant industry was really born with the rhizobia, and it continues to this day with rhizobia and legume inoculants. The arbuscular mycorrhizal and ectomycorrhizal inoculants have different sorts of histories. The ectomycorrhizal inoculum has been used since the 70s in the pine plantation replanting industry to great success. So they inoculate them before they outplant them. Then they have mycorrhizal associations when they go out, and it really, really helps with survival of those seedlings. So that's a big industry because it's easy, you can grow the fungi. But on the arbuscular mycorrhizae, those are usually for very specialized types of applications where you have, for some reason, killed off your mycorrhizae. Either you fumigated. So a good example is the citrus industry. They tend to fumigate, but citrus is very mycorrhizally dependent. So they found they needed to inoculate these things back in. But it's usually more specialized situations for that. And then you have these soups out there, and you can go down to Green Tree and take a look at the labels. And I'm thinking, they have rhizobia in here, but how do they even know that they're capable of fixing nitrogen? You know, it's so much is unproven about those microbial soups that I'm quite leery of them myself. So a specific organism designed for a specific task, we can have a lot of success with that.
Thank you.
Sure.
Since no one else is jumping in, I have another question. Korean natural farming, they kind of tend to focus on indigenous microorganisms that they can add to. Has there been any scientific literature about if they're effective or if they're actually useful in any capacity that you're aware of?
Interestingly, I'm just reviewing a chapter now out of India on these types of biofertilizers. So there is an organization, the Indian Association for Indigenous Agriculture in Chennai, and they have a huge collection of different types of approaches. They brew all kinds of things, not just compost teas, but all kinds of different fermentations there. They have things like cyanobacteria, azola, anabina symbiosis. So they maintain a lot of different things there. So yeah, there is a literature on putting together different types of fermentations for different types of purposes, and there is some success with that. We have not taken that approach here, but in the tropics, I've seen a lot of different applications that have been fairly successful.
Was there a test for the nitrogen?
In terms of the literature, I can give some to Joseph for you, Joseph Ancilli for you. And then you can have a look there on what's actually been productively used. But again, there's not a huge industry here in the US to my knowledge.
Thank you.